Hi, I would like to first thank to the BioHack Academy and to the Garagem FabLab for the invitation. My name is Andres. I am also known as Dom in the biohacker arena. I have been working more than 10 years with genetical engineering, molecular biology. I am part of an organization called Syntek Bio, which works with biohacking all around Latin America. And today I'm gonna talk about biohackers from the Latin American perspective. First question that comes to mind is what is a biohacker? Well, once you talk about biohacker, you're pretty sure we're talking about information. Biohacker, when you remember the hackers from computers, which are people that know a lot. And in biology, everything is information. If you think about your genome, the sequence of your genome, what we're dealing is with data and how to understand it. And in biology, we have people who wants to understand also biology and wants to hack it, wants to build stuff with it. We have different flavors of biohackers. For example, we have what we call grinder, which is the enhancement of the body. As you say in this first picture, a person who has a sensor in his right and left hand. And we also have the do-yourself biology community, which is more related to what I'm gonna talk about today. For the do-yourself biology community, one of the most important things is the open source. The open source movement is pretty important for us because it's a way to give tools to people to understand this technology, to relate to it, and this is the best way to change the world. We won't be able to do it by ourselves, but if we put in the hands of people the knowledge and the tools, together probably we will change a lot of things that we think are wrong. When you decide to become a biohacker, initially you don't have much idea of what you are doing. This is how everybody works and everybody begins to any discipline in the world. And then you begin to evolve, you begin to learn a little bit about how things work. So you become something like we could say a Windows user. Like you know how to do the stuff, but you're not pretty sure how it works. And finally, you get to the upper part of the screen when you are kind of advanced user, when you know how to use a terminal, how to write code, and that's why we call a biohacker. Normally, the people that call themselves biohackers at the beginning, they're more like a Windows user. This is because for you to be a real biohacker, you have to spend a lot of time in the laboratory. You have to spend a lot of time reading and studying because biology is pretty complex. And this is kind of a problem because you need like a PhD for developing or using this technology right now. So the question is how we can change that, how we can make more accessible this technology to people. Well, when we want to do this, we will face three bottlenecks. The first one is the knowledge, as we said. It's hard to learn this, but there are more open source journals and information all around the internet that can help with this. The second bottleneck is the hardware, is the equipment. This is you need pretty expensive equipment to develop this kind of activities, and that's why probably you are all here because the Biohack Academy with this class is trying to teach you how to build these machines by yourself using low cost materials, and in the process of doing that you will begin to understand how this works. So this is also a way to hack the knowledge because you learn how to build the machine, what to do with the machine, and afterwards how you can actually improve the machine through the time and make it cheaper and make it more accessible to other people. And finally, the third bottleneck is the chemistry. Even if you have the knowledge and the hardware, still there's a chemistry part that is still pretty expensive when we talk about enzymes and proteins that are pretty important for your work, but they are still expensive. That's why we decide to create a company that helps people to not care about the enzymes or all the machines so they can use this technology through the cloud. And this is a company that I'm right now co-founder, and we open it in United States. So I brought a slide from a presentation that I gave last year at Germany in the Falling Walls competition, in which I was explaining the idea. If you see on one side we have the first computer, one of the first computers, then this technology was locked down in universities, in big laboratories, and then we have the personal computer. This is not the first personal computer, but is one of the more famous. And the important thing about this transition is that we put this technology from the universities into hands of people, and they began to play with these, they began to create, and now we have amazing computers in our pockets, which are our cell phones, and these are more powerful than even the computers we use to take the man to the moon. So in the other side we have biology. Right now it's a pretty expensive and complicated technology that we have locked down in universities. The idea is to break this wall and make it accessible to everybody so the people can impact society the same way that computers did it with this technology. And the proposal was to use molecular automation, software and hardware, and user-friendly interfaces to make this real, to make it easy for people to use this technology. And I'm gonna give you a small example of what can happen when people have access to technologies like this. So we have, for example, the Arduino. We have the Arduino community and we have the MakerBot community. Both of them help us to learn how to prototype circuits. We want to do the same with biology. They also help us to print actually these ideas we have prototyping. We don't need more big companies to prototype ideas and print it out, because now we have 3D printed, which has been here for a long while, but now is really popular and a lot of people is using it from home. So what I want you to take home is to remember how the world can change once you put the tools in the hands of people. And this is what means technology democratization. Well, coming back for the context of biohacking, I brought this guy. I know most of you know who is him. Is Richard Feynman, one really well-known physicist, and he actually was talking about nanomachines, nanoparticulates, all around 1959. So this guy was 50 years ago already talking about nanomachines. And this, in the other image, what you are seeing is a ribosome which is reading DNA to produce protein. This is what happened in your cells. So he already knew, and he was already thinking about these kind of machines that we will use someday for probably try to tackle problems like diseases. And these machines are already inside of us working. This is actually a nanomachine processing information in your cells. So when we put all this information together, the 3D printer movement plus the biohacker movement, we can build amazing things, like this printer of organs. There's already a researcher working on this. He's called Anthony Atala, and he already actually print a bladder and is working towards printing a liver. And how it works? Well, you have a basic CNC machine with a USB microscope and a way to dispense the cells. And the part that is related to the biohacker is what they call the bioink, which are the cells, and we need to understand how these cells divide and how they grow and how they differentiate, so we can use these two technologies together to build organs. The process is not exactly 3D printed right now because he uses organ from a deceased person. He cleaned all the cells and then he used the 3D printer to put back the cells inside it. So the cells that he's using are from the same person that is gonna receive the organ, which means that this person won't have any problem, and he won't have to take immunosuppressors drugs which is the main problem in the transplants, because this will be built his by his physician using his own cells. And this is the second important idea I want you to take home. This is called technological convergence. And now I'm gonna begin to talk about how we got here, because we want to understand how the technology is going into this point that we can actually convert what is called 3D printed with biohacking, and how actually biohacking appeared. The first point we need to think about is the decrease in the prices of sequencing genome. So in 2011, in 2001, when they finished the first genome, it was around $100 million. Now in 2013, we are talking about that you can sequence a genome for less than 10K. And the price actually is going down, and we expect in the next 2 years we'll be able to sequence genomes for $1,000, which means that we're gonna get into the door of personal medicine. When we can sequence a human genome for 1K, we begin to understand diversity. We will begin to have real data to compare the diseases between one and another person, and this is pretty powerful. This happened, as you've seen in the graphic, because we have a really big decrease in the price that was even more than we were expecting if we modeled the decrease by the Moore's law. And this was thanks to the second generation of sequencing, where we have 454, Illumina, and another platforms that make this task pretty cheap. And I'm gonna give you a little example of how this works. If you see these genomes, you have Saccharomyces cerevisiae, which is a yeast. You have Oryza sativa, which is rice, and you have the Homo sapiens. And you see also the size of the genomes. So you have 12 million base pairs in Saccharomyces cerevisiae, 470 million base pairs in Oryza, and you have almost 3 billion base pairs in Homo sapiens, which means you have to read 3 gigabases. And now I'm showing to you, this is the big machine is a sequencer that uses Sanger for reading the genome, and the small one is the second generation. It's a second generation platform. So if we read the genome just once, and this is not how it works, because normally you read the sequence more than once for be sure about what you're reading, but let's say you read it once using Sanger with this machine, which is huge, it will take you around 10 years. Next to it is a second generation platform. This is the real difference on size, so the small one is around the size of a microwave. It can sequence 15 gigabases per day, which means in 5 hours you're gonna get one human genome sequence. And this is how this explains how disruptive was this technology, and how it was important, because the databases began to grow with crazy amounts of data. And the interesting thing is that this data is available to you. You can go online and just look into the gene bank and download the sequences, and you have other databases like the GOLD, the Genomes Online database, that you can access. You don't even need to be in a university. And you see that we have around more than 8,000 complete genomes that have been sequenced, and you have more than 22,000 genomes that are in the way to be sequenced. They are incomplete genomes. And then you begin to think how much information you can access and actually you can use. And the other tool that we have that was brought by what we call synthetic biology is abstraction, in which you put something so complex as biology, and this could not be the best way to do it, but it's an approach that we can use to try to tackle this complexity. You put it in different layers of complexity using abstraction. So in the first layer, you have the DNA, just sequence. The second layer, you have what we call a para, which means you already know what is the meaning of that sequence if it codifies for a gene like a a red fluorescent protein, or if it is codified for a control part like a promoter or RBS. So you have a part, and when you put the parts together, you build a device which will be a higher level of abstraction, in which you have an signal that is going into and an output, and then you don't have to take care more about the DNA sequence because you already did that in another part of the process, and then you put these devices together and you build a system which could be a metabolic pathway. So this is a way to approach biology to help us to tackle this complexity. Nevertheless, we still have these bottlenecks. As I explained it to you at the beginning, we had the bottleneck of the cost, because we need equipment, we need chemistry, we need knowledge. We also have, as I was just explaining to you, the bottleneck that are related to the technology, which is the complexity. And we also have tools like the abstraction that I just showed you, or standardization of the part, which is pretty used by the agent competition, or single cell resolution, or process modeling and automation to understand all this complexity in biology. For example, we are Syntek Bio, we are working toward reducing the complexity and the cost. And the cost, it means we use first molecular automation, which is a concept that help us to build experiments and to build these applications with less steps and also with less cost. These are parallel projects. And also we are supporting the participation at the agent competition, which helps students to learn about synthetic biology and engage into research. And why this is important? In the future, we will face big problems, and we know that because our population is growing. It's supposed to be around 9.5 billion people by 2050. So we need to produce more food, we need to produce more energy. Probably we will need to explore other planets and even try to build some ecosystem up there for us to live. And this is not me talking you and making this up. This is coming from the NASA organization. This document has at least 5 years where they began to study this technology as a possible tool for making this real, for help us to go to other planets and establish ourselves in there. What are the examples of applications? Well, we have the case of the artemisinin, which is a drug that is normally used against malaria, but malaria normally it's found in populations that they don't have much resources, so it's really a drug that should be not expensive, but the problem is that as we produce right now, it is pretty expensive because it's produced by a plant, so you have to grow the plant, extract the compound, and this population they are not a position that they can afford that. So Jay Kislik took the pathway to produce artemisinin for a plant and put it into a yeast, and that really reduced the cost. And just last year they began the industrial production of this drug. After almost 8 years they figured out how to do this on the bench, on the laboratory. Another example is the picture that you're looking at, which is the production of bioplastics. So the shapes are bacteria, and the white shapes inside of it, the circles, are actually bioplastic production. So it means that we can produce new materials that are more environmental safe, and this is why we need to understand this technology. There's also people that is working with these technologies using in a really amazing ways. So if you see in here, we have examples of companies like the one in the right corner, which you send your DNA and they will run it into a gel, take it a picture, and then they're gonna send you that picture of your DNA digested by restriction enzymes, and you can put that at your room. The other companies which are building kind of base tattoos that you can program if you are not into much in the idea of inserting one of those in your hand as the first picture. Maybe you can just stick it into your skin, like in the second picture of the MC10 company. And there's also an artist like Susan Lee, which actually used bacteria to produce a tissue, and with this tissue she made clothes. She made this actual jacket that you are looking at. There are also older companies like this one, Clove Hitch, which comes from 2003 when they introduced this product, which is a fish that is genetical engineering. So you have the fish with the expression of these different proteins that make them glow red or green. This is, as I'm telling you, 10 years ago. This is not so new as you might think. And finally, this artist, which did an experiment. Instead of taking boxes, his first installation was taking boxes with things of his friends, personal things that he can store. And then instead of that, he built this vending machine, and each of these boxes, instead of having a thing that come from his friends, actually had DNA that he extracted from his friends. So they make like a party and all the friends went there and they did a DNA extraction of their own DNA, and then they give it to this artist so he can do this installation in which he has his friend's DNA material into boxes into a vending machine, which is pretty cool. Finally, I would like to thank to the people that is in Syntek Bio. We have 4 projects right now running, and thank you.